Welcome to Microbiology classroom. Today I want to discuss about the bioremediation, how microorganisms are used for decomposition, degradation of the recalcitrant xenobiotic compounds. Bioremediation part 2. The pollution of soil takes place by different compounds. Pollution of soil occur due to heavy metals, pesticides, insecticides, herbicides, petrochemicals and other compounds. Water bodies are also polluted by the industrial wastes, heavy metals, oils, aromatic hydrocarbons, aliphatic hydrocarbons, etc. Metals, pesticides, herbicides, solvents, petrochemicals and all other contaminants are more prevalent forms of contamination found at water bodies, soil, waste sites, and their remediation is very difficult. There are different types of contaminants, pollutants. They include solvents, pesticides, herbicides, fungicides, insecticides, petrochemicals, explosives, polychlorobiphenols, phenols, chlorophenols, heavy metals, etc. Naturally occurring bioremediation and phytoremediation have been used for centuries. Naturally occurring bioremediation is used for cleanup of the environment and desalination of agricultural land by phytoextraction technique using the plants. The desalination of agricultural land was done from several centuries. Bioremediation technology using microorganisms was first reported by George M. Robinson. Bioremediation technology using microorganisms to remove the pollutants was first reported by George M. Robinson. George M. Robinson was the assistant county petroleum engineer for Santa Maria, California. He was assistant engineer at Santa Maria, California, petroleum engineer. During 1960s, he spent his spare time experimenting with dirty jars and various mixes of microbes. So he carried some experiments to remove the petroleum wastes from the jars using consortium of microorganisms, microbial mixtures. However, the first application of bioremediation for treating oil spills and anthropogenic compounds that began few years ago, 30-40 years ago in 1975 by R.L. Raymonds. R.L. Raymonds, the first application of bioremediation for treating oil spills and anthropogenic compounds began some 40 years ago with a report entitled Beneficial Stimulation of Bacterial Activity in Groundwater Containing Petroleum Products by R.L. Raymond and co-workers in 1975. R.L. Raymond and co-workers in 1975, they reported the bacterial activity against the oil spills, degradation of oil spills by bacteria. Raymond and his colleagues reported that addition of certain nutrients to subsurface soil increases the number of bacteria and bacterial activity in degradation of the hydrocarbons derived from the petroleum. So the nutrients added to the soil promoted the growth of bacteria and enhanced the activity metabolic activity of the bacteria and completely degraded the hydrocarbons of petroleum, thereby they completely removed the contaminants from the polluted site. This was the origin of the process of bioremediation. So this is known as the in situ bioremediation. So enhanced in situ bioremediation. So this was first done by the R.L. Raymond and his and co-workers. So R.L. Raymond first developed this in situ bioremediation process. So addition of certain nutrients to the contaminated soil to promote the growth of the bacteria, to degrade hydrocarbons derived from the petroleum. So this in situ bioremediation strategy, this was first reported by the Raymond and co-workers. So in this strategy, certain nutrients are added. In this strategy, certain nutrients are added to the contaminated site. So to the soil and groundwater, some nutrients are added. So these nutrients promote the growth of microorganisms. So these microorganisms readily degrade the complex xenobiotic compound such as these hydrocarbons. So addition of phosphate and addition of nitrate and addition of the electron acceptors such as nitrate and nutrients like phosphate and nitrogen source, addition of nitrogen source. So these things promote the growth of microorganisms, promote the growth of bacteria and increases the metabolic activity of the bacteria, so that the hydrocarbons derived from the petroleum are readily degraded. So this process can be applied to the subsurface soils. It is also applied to the water bodies such as lakes and other bodies of water. The biochemical capabilities of microorganisms seems almost limitless. Microorganisms are its greatest chemists. So microorganisms can be used to remove the xenobiotics and other complex organic substances, hazardous substances from the sites, from the contaminated sites. Bioremediation is the cost effective and only one method available to remove the xenobiotics and hazardous substances from the contaminated site. Degradation of the pesticides. Pesticides are the agrochemicals used to control insects or pests on the crops. Pesticides, these are agrochemicals used to control insects or pests on the crop. These pesticides are sprayed or applied on the crop. So only 0.1% of the pesticide sprayed is used to kill the target pest. Remaining 99.9% spreads into environment and into the food. So 99.9% sprayed pesticide remains in the environment and food. Only 0.1% pesticide is used to kill the target pest. So the pesticide which entered into the environment, which entered into the food, that becomes the problem. Pesticides are classified into different types. So pesticides are different types. Pesticides are categorized into different types. Bactericides kill bacteria. Fungicides kills fungi. Insecticides kill insects. So nematicides, they kill nematodes. So pesticides are different types. They are bactericides, fungicides, insecticides, nematicides. So pesticides are categorized into different types: bactericides, fungicides, insecticides, nematicides, herbicides. So fungicides kill fungi, insecticides kill insects, nematicides act against nematodes, herbicides they kill herbs. So pesticides, They are categorized, classified in other manner into different types. So pesticides are categorized into pyrethroids, organophosphorus compounds, organochlorades, derivatives of carbamic acid. So organic pesticides are categorized into following types. First one, pyrethroids. Second one, organophosphorus compounds. Third one, organochlorades. Fourth one, derivatives of carbamic acid. Pyrethroids are natural pyrethroids extracted from plants. Synthetic pyrethroids, they are similar in structure to natural compounds. They are synthesized by chemical synthesis. Examples include pyrethrum, cypermethrin, phenolate, etc. Organophosphorus compounds, these are derivatives of phosphoric acid. Derivatives of phosphoric acid. Example, malathion, parathion, dichlorophos, bromophos, phentheon, cyanophos, trichloromethophos, pasolone, trichlorophene, etc. Organochlorades. Organochlorades are, these are chlorinated hydrocarbons with one or more benzene rings. Chlorinated hydrocarbons with one or more benzene rings, organochlorades. Example, DDT, BHC, lindane, heptachlor, dichlor, aldrin, endrin, dieldrin. So these are examples to organochlorades. Derivatives of carbamic acid, these are another category of pesticides. These are derivatives of carbamic acid. Example, carboryl, primicarb, begon, benthanol, thiram, gyram, manab, ginep. These are the derivatives of carbamic acid. Several factors influence the fate of pesticides in soil. So the fate of pesticides in soil, that is influenced by several factors. What are these factors? Number one, chemical decomposition. Chemical decomposition of the pesticide. Decomposition of pesticide, this is one. Second, photochemical decomposition. Third, volatilization. Fourth is movement in soil, plant uptake, adsorption, microbial decomposition. So there are several factors influencing the fate of pesticides in soil. Majority of pesticides, they have a hydrocarbon skeleton with different substitutions. If the substitute group is halogen or nitro group or sulfonate group, the persistence of pesticide will increase. Presence of halogen, chlorine, or presence of nitro group or presence of sulfonate group on the hydrocarbon is responsible for persistence of that pesticide. Methyl, methoxyl, carboxyl substitutes cause moderate persistence of pesticide, whereas presence of halogen, chlorine or nitro or sulfonate group makes that pesticide will remain for very longer time. Organochlorades with chlorine group will have long persistence than other pesticides. So presence of the chlorine group, that makes the long persistence of the pesticide. More extensive the chlorine, in general, the more extensive the chlorine substitution, the more effective the pesticide, but it persists in soil for long time. If number of chlorine molecules are more in the pesticide, that pesticide is more effective, but it will remain very longer time in the soil. For example, 2,4,5-T trichlorophenoxyestic acid is more resistant to biodegradation and persists in the soil than 2,4-D, 2,4-dichlorophenoxyestic acid. 2,4-D is biodegraded in 3 months, while 2,4,5-T requires 2 to 3 years for degradation. So with that you can see the effect of chlorine. So more number of chlorine atoms are there in the herbicide or pesticide, so its decomposition is very slow. 2,4,5-T degradation decomposition require 2 to 3 years, whereas 2,4-D where only 2 chlorine atoms, it is biodegraded in 3 months. So additional chlorine in 2,4,5-T interfere with the hydroxylation and cleavage of aromatic ring, so that 2,4,5-T persists for very long time. Pesticides and their period of persistence. Organochlorates like DDT, the period of persistence is 21 years, heptachlor 16 years, chlordane 5 years, BHC 3 years, organophosphates, diazonium 3 months, disulfotone 4 weeks, porate 2 weeks, parathion up to 2 weeks. This is the period of persistence. DDT 111 trichloro 2,2-bis 4-chlorophenyl ethane. So this DDT is a xenobiotic pesticide. It is persistent for very long time in the soil. DDT is prohibited. Usage of DDT is prohibited in several countries. However, DDT is used in few countries. DDT is a persistent environmental pollutant. It undergoes slow degradation in environment. The steps involved in degradation of DDT are reductive dechlorination of trichloromethyl group of DDT to form 2,2-bis 4-chlorophenyl ethane DDD. So the DDT then undergoes dechlorination, oxidation and decarboxylation to form bis 4-chlorophenyl methane DDM. DDM again undergo cleavage of aromatic rings to form para-chlorophenyl acetic acid, which is finally degraded to CO2 and water. Alternatively, DDM may be converted to DBH 4,4-dichlorobenzohydrol or DBP 4,4-dichlorobenzophenone. DBH and DBP also undergo reductive dechlorination of the aromatic ring to form non-chlorinated compounds, which then undergo aromatic ring cleavage and degradation. You can see the degradation of DDT, how the DDT is degraded. 2,4,5-T is also degraded. 2,4,5-T is degraded first to 2,4,5-trichlorophenol, then it is degraded to 2,5-dichloroquinone, then it is converted to 5-chloro-2,4-dihydroxybenzene. It is converted to 1,2,4-trihydroxybenzene, then mallyl acetate, finally 3-oxoadipate. 3-oxoadipate is degraded to succinate and acetate. Then succinate and acetate is also degraded to CO2 and water. Burkholderia cepacia and Pseudomonas species, they degrade the 2,4,5-T. Burkholderia cepacia, it readily degrades 2,4,5-T. Burkholderia cepacia uses 2,4,5-T as sole source of carbon and energy. Bacteria that can degrade the DDT aerobically include Bacillus cereus, Bacillus subtilis, Escherichia coli and Enterobacter aerogenes. Some of the fungal genera degrading DDT include Mucor alterans, Aspergillus flavus, Pleurotis versicolor, Pleurotis austriatus, Gliophyllum trabeum and Phyllius vieri and Phanerochaete chrysosporium. Petroleum products, oils, they are sometimes leakaged out from the tankers or due to accidents, etc., and drilling accidents. Oil spills are formed in the water bodies. Oil spreads in the water bodies. This oil, petroleum products and oil is insoluble in water. Petroleum hydrocarbons, they are deficient in essential elements like nitrogen, phosphorus. So due to non-availability of this nitrogen and phosphorus in petroleum hydrocarbons, microorganisms do not readily degrade the petroleum. Petroleum hydrocarbons, they are not readily degraded by the microorganisms due to non-availability of nitrogen and phosphorus, and moreover they are insoluble in water. As a result, their degradation becomes problematic. So bioremediation of oil spills is greatly enhanced by the natural residential bacteria by addition of fertilizer containing nitrogen and phosphorus. Nitrogen and phosphorus fertilizers are added to the area where oil spills are present. So this addition of nitrogen and phosphorus greatly promote the growth of residential bacteria, and these bacteria, they decompose the oil spills. The Gulf of Mexico spill of 2010, a deep water drilling accident, showed that the chemical dispersants that added to the oil spills broken the oil spills into microscopic droplets. So dispersion of the oil layer into microscopic droplets enhanced the degradation of these microscopic droplets by the microorganisms. So the Gulf of Mexico spill of 2010, a deep water drilling accident that caused a spread of the oil layer to the several miles, where scientists, they used the chemical dispersant. They sprayed the chemical dispersant to disperse the oil layer into microscopic droplets. So when this oil layer is dispersed into microscopic droplets, microorganisms readily degraded these microscopic droplets of oil. Compared to earlier Exxon Valdez tanker disaster in Alaska, dispersant treated oil has been metabolized rapidly in 2010 in Gulf of Mexico. So there are some reasons for this. Temperature of Gulf is warmer, and temperature in Alaska is very cold. This is one reason. So for rapid degradation of the petroleum spills in Gulf compared to Exxon Valdez tanker incident in Alaska. From that, usage of the dispersant, chemical dispersant that broken the oil into microscopic droplets at Gulf of Mexico spill in 2010, that enhanced the microbial degradation of the microscopic droplets of oil, so that the bioremediation occurred very quickly. Hydrocarbons present in the petroleum contaminated site are degraded by the microorganisms in two conditions, oxic and anoxic conditions. Hydrocarbons are degraded by microorganisms in oxic and anoxic conditions. In oxic conditions, oxygenases introduce oxygen into hydrocarbons. In oxic conditions, oxygenases are the enzymes which introduce oxygen into the hydrocarbons. Oils are insoluble in water and they had less density compared to the water, so oils float on the water. Hydrocarbon degrading bacteria attach to the oil droplets and decompose oil and disperse the slick. Alkani Borax barkumensis Alkali Borax borkumensis grow only on hydrocarbons, fatty acids or pyruvate, produces glycolipid surfactants. So glycolipid surfactants produced by alkali borax that break up the oil into small microparticles. So these are readily degraded by the alkali borax barkumensis. Under ideal bioremediation conditions, up to 80% of the non-volatile components of oil can be oxidized within a year of oil spill. Under ideal conditions, ideal bioremediation conditions, suitable temperature and suitable aeration, ideal conditions, up to 80% of non-volatile components of oil can be oxidized within a year of oil spill. Anaerobic hydrocarbon oxidation is carried by denitrifying bacteria, sulfate reducing bacteria and phototropic bacteria. Anaerobic hydrocarbon oxidation is carried by denitrifying bacteria and sulfate reducing bacteria and phototropic bacteria. So these organisms are responsible for hydrocarbon oxidation in anaerobic conditions. Toxic catabolism of aliphatic hydrocarbon hexane occurs by addition of fumarate to hexane. Addition of fumarate to hexane provides oxygen atoms necessary to form fatty acid derivative that is catabolized by beta oxidation to yield acetyl-CoA. Electrons and hydrogen generated from hexane reduce sulfate or nitrate in anaerobic respiration. Oil spills, hydrocarbons, aromatic hydrocarbons, aliphatic hydrocarbons, these are readily degraded by the bacteria Pseudomonas putida, Dichloromonas aromatica, Dinococcus radiodurans, Methylibium petrolipylum, alkali borax borkumensis, Phanerochaete and the fungi Phanerochaete chrysosporium and Methylibium petrolipylum. So the bacteria include Pseudomonas putida, Dichloromonas aromatica, Dinococcus radiodurans, Methylibium petrolipylum, alkali borax borkumensis and Methylibium petrolipylum. These are the bacteria readily degrading the oil spills hydrocarbons. Using fungi as a potential treatment of contaminants began in 1985 when the white rot species Phanerochaete chrysosporium was discovered to metabolize a number of environmental pollutants. Phanerochaete chrysosporium was the first fungi linked to degradation of organic pollutants. So this fungus possesses tremendous potential for bioremediation of pesticides, polyaromatic hydrocarbons, dioxins, carbon tetrachloride and many pollutants. Phanerochaete chrysosporium has become the model for bioremediation. Other notable species of white rot fungi include Pleurotus austriatus and Trametis versicolor. Fungi which are used for bioremediation of hydrocarbons include Trametis versicolor, Chrysosporium lignorum, Pleurotus austriatus, Coriolapsis polyzona, Pleurotus sordida, Coriolus versicolor and Phanerochaete chrysosporium. So what are the conclusions? Bioremediation has emerged as a powerful tool, as an integrated toolbox for environmental cleanup and ecosystem service provider. So bioremediation, it has emerged as a integrated toolbox for environmental cleanup and ecosystem service provider. Bioremediation has tremendous potential to provide a low cost, non-intrusive natural method to remove the hazardous substances, toxic substances in soil, water and contaminated sites. Actually, currently research is being conducted to improve and overcome limitations that hinder bioremediation of pollutants. On a broader scope, much research has been and continues to be developed to enhance understanding of the essence of microbial behavior as microbes interact with various toxic contaminants. Additional research continues to evaluate conditions for successful introduction of exogenic and genetically engineered microbes into a contaminated environment and how to translate success in the laboratory to success in the field. Thank you for listening the bioremediation